The overall goal of this procedure is to determine the glycogen content in cyanobacteria using an enzyme-based selective hydrolysis and assay.
This method can help answer a key question in the cyanobacteria field, such as physiology, molecular genetics, and bioengineering of different cyanobacteria strains under investigation.
The main advantage of this technique is that it's adapted to a small scale, is easy to perform, and highly sensitive and specific to glycogen.
Though this method can provide insights into cyanobacteria, it can also be applied to other microorganisms that accumulate glycogen or starch, such as E. coli, yeast, microalgae, and various heterotrophic and phototrophic microorganisms.
Begin this protocol with preparation of cyanobacterial cultures as described in the text protocol. Transfer 1 milliliter of cyanobacterial culture or cell suspension to a 1.5 milliliter tube. Then, centrifuge at 6,000 times G and 4 degrees Celsius for 10 minutes. Discard the supernatant before resuspending the pellet in 1 milliliter of 50 millimolar tris HCl pH 8. Centrifuge the resuspended pellet as before. Discard the supernatant and resuspend the cell pellet a second time in the tris HCl buffer. Centrifuge the tubes as before and discard the supernatant. Then, thoroughly resuspend the pellet in 500 microliters of 50 millimolar tris HCl buffer pH 8. It is crucial to have the pellet well suspended for an efficient lysis.